All right, very good. Well, welcome everyone. Thank you so much for attending the November Jam meeting. It's great to be speaking again. This is my second time speaking to Jam, and I'm quite excited to get to share the work that we've been doing at BPHL when it comes to SARS-CoV-2 sequencing, and not just the sequencing. I think the meat of the talk is really going to be about what happens with the information after we sequence it. But before we get to that, you're going to hear from me, and we're going to go over how we go about sequencing SARS-CoV-2. So we're going to start with the wet lab portion, and then we'll go into data analysis, data submissions, and a really cool visualization of the strains via NextStrain. So we're going to start with the wet lab protocols, and I am Matthew. So with SARS-CoV-2 whole genome sequencing, there are 4 major steps that have to be considered when sequencing an RNA virus such as SARS. Each one of these steps has its own challenges that you have to overcome. And early on, while we were developing our process for SARS-CoV-2, each one of these steps was really just a challenge. Just getting cDNA from a extraction was a challenge, but some strains, like to convert better than others. That's in the next step, enriching for SARS-CoV-2, doing the amplification, another step where something can go wrong. Sequencing, we'll go into that a little bit more later in the presentation, and then how we assemble the genomes. So the generation of a sequence at BPHL has many steps where the sample can be denied or the data is just not good for downstream analysis. So we start with all of the samples from Florida that come into the Bureau of Public Health labs. We are obviously seeing mostly county health department samples, but we also see from hospitals as well. And then from there, we have to do our clinical testing, our real-time PCRs, and to see if the sample is positive or not. Once that is done, we usually take the eluate from those runs, usually on a Abbott M2000 or a Hologic Panther. I guess we're bringing on the Cobus, but we can't save the eluate from that, so never mind. And then from there, once we have that stored and frozen and cataloged, then we can go in and take the extracted DNA and go and sequence it. Once we do the sequencing, we have to see if the quality of the sequence is good enough for downstream analysis. And then from there, did they pass QC? And Sarah will talk more about what happens with the data after that. So the timeline that we went through when we were developing our protocol for sequencing SARS-CoV-2 started around March 2020. It was our first attempt at sequencing COVID, and we wanted to target the iSeq platform by Illumina. It's a really convenient and small platform. It's got one of the lower throughputs of sequencers that Illumina provides, but when it comes to small genomes, the iSeq is usually a good place to start. We started developing our protocol with knowledge that we had gained developing an HIV sequencing protocol. So a lot of the information that we attained when it came to what was good reagents for converting the cDNA for amplification. So we kind of had a backbone to go off of. And then when we were able to first sequence it, we ran comparisons on almost all of the Illumina sequencers, all the ones we have at least, multiple MySeqs, an iSeq or 2. We have 2, I think we tested it on both, and the NextSeq, which is the highest throughput that we have. We also had Taj Azarian down in UCF test on the MinION during doing those parallel runs. And then in April 2020, we scaled up our sequencing efforts to utilize the MiSeq as our main platform. So in March, we were targeting the iSeq. In April, we went ahead and scaled up. And we were going through a protocol that was really time consuming. We were using a prep kit called the Kappa prep kit, which I'll go into more later. Also, the V2 primer set was introduced, which I'll go into more later as well. And then May 2020, the MiSeq and Nextera XT from Illumina, the library prep kit, which I'll talk about here coming up, becomes standard, and that's how what we use today. So pivoting a little bit, the Arctic network is the basis and publicly accessible repository for laboratory protocols, bioinformatics tutorials, data sets, primers, all the data that we need to support SARS CoV-2 sequencing is gathered and at the Arctic network. Here, I have the website if you'd like to go and view it. They consider themselves a real-time molecular epidemiology unit for outbreak response. And the project is developing an end-to-end system for processing samples from viral outbreaks to generate real-time epidemiological information that is interpretable and actionable by public health bodies. And that's really the kind of the thesis of the talk, is that how do we make this data interpretable and actionable for the public health bodies? So Florida's laboratory developed protocol utilizing Arctic primers. Usually going through protocols is a little boring, but you know, I gotta lay it out. So the RNA is sourced from the real-time PCR platforms that we have in-house. I discussed the M2000, the Hologic Panther. The supported transport media include saline or VTM. The RNA is then converted to cDNA by utilizing reverse transcriptase enzyme. We use the superscript 4 from Thermo Fisher. Then when we amplify our product, we use the Q5 master mix from New England Biolabs, and we use the version 3 primers from the Arctic network, which those are actually sourced by IDT. And I'll talk about it more coming up, but the reaction when it comes to amplifying our product is a split reaction, because the primers are separated into two pools. So when we're done doing that split reaction, we bring the samples together at equal volumes and dilute to 0.2 nanogram per microliter. Then we go right into the Nextera XT protocol, which is from Illumina, and I'll be going over that as well. So the Nextera XT protocol by Illumina is a really great library prep kit, and it's something that we've been using in the PulseNet lab since the beginning. And they've iterated on it and made the Nextera Flex kit, and it's just really done well, to the point that we're still using the XT kit, which is what we started on back in 2016. So it's a convenient kit for us to use because it is something that we have in-house. So the Nextera XT kit does enzymatic cleaving and adapter ligation all in one step. It's the enzyme is called the transposase. And then from there you do universal dual indexing. This is something that was developed later on. Usually the indexes come in two separate pools, and you have to add them separately, but the universal dual indexing kits have come out and made it to where the indexes are paired up. And then after that, you just do a bead-based cleanup, pool all of your samples together, and sequence. So I mentioned these Arctic primers, and they're really, really cool and really unique. It's two pools of primers that require separate reactions. So you get your really nice cDNA. You then amplify it in two different tubes. Let's just say pool A and pool B. You're going to get a tiled amplicon setup. And these are kind of short amplicons, around 400 base pairs, and they alternate between pool 1 and pool 2. There are 98 amplicons generated between the two reactions. And we're going to get near entire genomic coverage when it comes to the Arctic primer pools. Now, these have iterated over time. They started with version 1, and then they went into version 2, and now what we use, which is version 3. I'm going to go over a little bit what has changed and to show you visually a little bit of how you can tell where some areas of the genome were dropped out in version 1, and then they show up in version 3. So right here, we're going to look at some dimer formation that occurs in amplicon primers 18 and 76. We'll look at that in 1 second. So here's just a depiction of the alternating primers. It's not the best image, but it was the best I could find. You just have to think of them as alternating, and the first one being in pool A, second one in pool B, and so forth. So this is a depiction of version 1 primer pool on the top and version 2 on the bottom. And you can see that when they fixed the dimer formation at location 18 and 76, that in the B chart, you can see how the coverage has now come back up to around 100x. Whereas before they were almost completely dropped out. So that was something that they really focused on, is how to get these dropout regions to pop tall when it came to the final outcome. This is a depiction between version 1 on top and version 3 on bottom. You can just take a look at it for a minute. There is a lot of areas that have been solved, a lot of areas that have not been solved, but it kind of shows you what they're trying to do when it comes to increasing coverage across the whole genome. So previously I mentioned the sequencing platforms. Here's just a image of all three of them: the iSeq, the MiSeq, and the NextSeq. And the iSeq is a small, cute little guy. It's about the size of 2 shoe boxes, and it does 24 samples on their version 2 chemistry. About 24 hours to run the iSeq. The MiSeq is a much bigger, still desktop or tabletop, but a much bigger unit. It does 96 samples in 48 hours, and we run that on Illumina's V3 chemistry, which is has a higher throughput than their version 2. And then on the NextSeq, we can do up to 384 samples on our mid-output kit. It runs really fast for that many samples, 26 hours. It's just a little bit too high of throughput for our needs at this moment. So this is a run metric that comes off of a MiSeq, since that is the platform that we are using going forward. I thought that I would just show you real quickly what it is that we look at when we look at a sequencing run. What information do we see here that tells us, is this a good sequencing run? Will this data be good coming out the back end going into Sarah's pipelines? First thing we'll look at is on the upper right, the Q-score distribution. And we'll see that overall it's a 90.5 Q30, which means that it's really high quality data. Down in the middle, lower, data by lane, It's clustered around 1,000, which that's a metric that's specific to the run. And you just want to get anywhere between, I don't know, 700 and 1,000. And this run is really perfect. You can also see on the intensities, the data by cycle. It shows the intensities of A, C, G, and T. Nice and smooth, and on the heat map, there's not a lot of noise. So this is going to be a really good run. The sequences are going to be really well done. So last but not least, some things that we've tried. We've tried a one-step superscript master mix, which is the reverse transcriptase mixed with a heat-blocked TAC. And this was just not giving us the yields that we needed. So we went to a dedicated reverse transcriptase and a dedicated TAC polymerase. The kappa RNA hyperprep kit was the kit that was being referenced as the gold standard for sequencing SARS-CoV back in the beginning. This kit was really cumbersome. You had to do your end conversion and then your DNA adapter ligation separate, and then you had to do cleanup, and then you had to do post-ligation cleanup. It was really just a mess. So we moved to what we had in-house, which is Nextera XT, and the quality has been great. We were using the NextFlex dual index DNA barcodes coupled with the kappa RNA hyperprep kit. So we moved on to the Nextera as well there. We moved on from V2 Arctic primers to the V3, and I showed you what happens to the coverage across the genome with the new primers. And then we also attempted tailed amplicon, but everything that we tried has been replaced with Illumina's Nextera XT, and everything has been going really well. All right, well, now I have to stop sharing.
Okay.
And then go to Sarah, make host. I think it's all you.
Okay, can y'all see my screen? Yes?
I can see it.
You can see it, okay.
Yes.
Great.
Yeah.
Want to make sure. Let me put it into, there we go, presentation.
There you go.
Great. Well, I'm Sarah Schmades. I am the bioinformatician at BPHL, and I also serve as the bioinformatics regional resource lead for the Southeast region that we help oversee expanding bioinformatics capacity to the different states. We have 6 other states and 1 territory, Puerto Rico in our region for the CDC AMD regions that we help out with their bioinformatics. So as Matthew went over the wet lab protocols, I'm going to dive into the data analysis and visualization, what we do with that data once we get it off the sequencer. I'm going to stick mainly to the slides, but I'm going to cut away in a few slides to do a quick little live demo in a little bit. So as Matthew mentioned, we're using the Arctic V3 primers to enrich for the SARS-CoV-2 genomes, or for the cDNA from those clinical samples. And what we end up with are, we're doing paired end sequencing runs on the MIC-seq or the NextSeq. And that's what we get off the sequencer. And then from there, we are then passing those FASTQ files into our in-house pipeline that I built. And we're running that on Hypergator. So if you're not familiar, Hypergator is the supercomputer at the University of Florida. It's a high performance computing cluster. This allows us to utilize high performance computing for our data analysis. We can do things in a timely manner and analyze a lot of samples all at once. And then from there, we can look at that data using different visualization methods and then go into the interpretation and submitting that to public databases. So our FLAC SC2 pipeline, that's the acronym for it if you didn't see it on the other slide, stands for Florida Assembly Quality. since we're generating assemblies, and then SARS CoV-2. So we're starting with our paradigm fast cues from the sequencer. And then after that, we go through a QC step. So with this, we are trimming any low quality bases and filtering out any reads that are below a particular length that are too short for our analysis, and doing that initial read pre-processing. Then after that, we are mapping those reads to the SARS CoV-2 reference genome. Most people are using the first genome sequence that came out of Wuhan. It's just referred to as the Wuhan reference genome, mapping those reads to the reference. And then from there, doing some additional QC, such as trimming away the primer regions, since we don't want to call variants from exactly the primer regions, but since those amplicons do overlap, we still do get the sequence over those regions, so we can call the variants, but making sure to trim away any sequence that would introduce any bias or known errors that would get introduced. And then reformatting some files. And then from there, we do variant calling. And I've built in some flags into our pipeline that will flag any samples for manual review that have any indels or have any particular mutations that may be causing a phenotypic change, such as early stop codons, so that we can manually review those to make sure that those calls are actually accurate before we submit that data. And then from there, we generate a consensus assembly. So we're basically at the very end of the pipeline getting one FASTA sequence. That is the 30 kb genome that we're going to submit. And then at the end of the pipeline, it also generates a report. So all of the samples that we've analyzed at once, it'll report out all the different assembly and sequence metrics for that run and for those particular genomes. So this is just an example of the first step. I'm going to go into each of the steps just in a little bit more detail, just to explain them a little bit more. So as I mentioned, with this first read pre-processing step, you're going to have some lower quality bases, mainly at the ends of the reads, that you want to trim away and then filter out any sequences. And so this is just an example here. This is actually a pretty good sequence, pretty good sample. But as you can see, just looking at the image on the left versus the image on the right, you kind of want everything in the green, which we're mainly seeing. But as you can see over there on the end, on the x-axis, you can see the position in the read sequence. So you see all the way over, we're doing 2 by 250 read lengths for sequencing. After trimming, you can see that variation at the end of the quality shrinks. And so we're left with extremely high quality data moving forward. So the next step in the pipeline is to then map those reads to the reference genome and call those variants. So as I said, we're using the Wuhan reference genome. It was the first sequence genome that was sequenced from in the initial days of the pandemic, or I guess before it was a pandemic. And this is just a screenshot just kind of showing you what that looks like. When we're at the top, you can see this is just a zoomed in shot of the reference sequence at the top. And then all the gray bars down below, those are all the individual sequencing rates. And then any of like the little colored lines in there, those are different variants of where the sequence differs from the reference genome. You can see it's kind of speckled. That's actually an example of different sequencing or PCR errors in there that get introduced, but they don't get called if they're not at a high enough frequency. That's how we can distinguish them between real variants versus not. So as you can see, the over on the right side, I don't know if you can see my mouse or not, but over on the right side, you can see the red bars that go all the way down. That's a real variant. That's a real SNP that's there. And that's how you can tell the difference. I don't actually look at all of them like this in this program for every single one of them, but this is what I use. I use this program called IGV when I am manually reviewing the samples to be able to try to decipher between a real variant and or a PCR or sequencing error. So the next step in the pipeline is to, and this is kind of the final steps, is to generate that consensus sequence. So what we're left with is about a 30 kb genome in a FASTA sequence. The way we've written the pipeline is that it will incorporate the major allele into that assembly. So we do see cases where you might have a mixed infection. It's considered standard in this field, and how you do your phylogenetic analysis is to generate just one consensus sequence using the major alleles. Anywhere where we have sequencing depth less than 10x, we use an N. We don't call that base. You don't want to call the reference genome, and we don't want to introduce a gap artificially. So we just call it an N. And then the pipeline will flag samples as either it passed QC or it failed. We do pass QC is it must have at least 80% genome coverage compared to the reference genome, and at least 100x mean read depth across the genome as well. Otherwise, we don't submit that sequence, and we don't include it in our downstream analysis. So then I'll then review any samples that were flagged for manual review. Most of these are samples with indels. We're looking at indels greater than 25%. We see a lot of, in like one base pair insertions, we see a lot, and these are just sequencing errors. We'll see some premature stop codons. Most of those are actually real SNPs in different genes, but we want to verify those so that we're not submitting anything that is maybe an error. And then at the end, we'll review the report and look at all of the assembly metrics from all of those samples. So after that, after we've run everything through the pipeline, we then have a FASTA sequence, a whole genome for each of the sequences or each of the sequences that passed QC. So the standard and what everyone is doing in the community is submitting those sequences to both NCBI, so to GenBank, the FASTA sequences to GenBank, the raw FASTQ files to the sequence read archive, as well as some people are submitting their alignment or mapping files to the sequence read archive as well, just so that others in the community can see how people created their assemblies. And then the other database is called GISAID. If you're not familiar with it, it stands for Global Initiative on Sharing Avian, or all started out for avian influenza. Now it includes all influenza data. And this is a separate database. It's a partnership with the German government and several other collaborators, and it's different than NCBI in that with NCBI, once you submit your data, it's publicly available to everyone, with the exception of if you put like a timed release or something on your data after publication. But with GISAID, the data stays in the ownership of the submitter, of the person that submitted their data. You also have the option to be able to update your submissions. So if you discovered an error or you want to update a sequence, if you got better coverage from a repeat sequence, you can go ahead and just replace that sequence. And they also have different data usage policies compared to NCBI. A lot of people want to use GISAID more so than NCBI. But the reason why I bring up GISAID and the reason why most of the scientists around the world that are doing this work and submitting their sequences are submitting them to GISAID, because one of the programs that we use called NextStrain actually pulls the data from GISAID. It's just easier to pull it from there. And so that's one of the advantages to using this database. So just to give you some numbers before I get into the data visualization. So these are our numbers of samples that we sequenced and submitted. And these are as of I have indicated on there, November 5th. So between, we did our first sequencing run for SARS CoV-2 on April 21st. Between April and then the end of October, we've sequenced over 3,800 samples at BPHL. Over 2,400 of those have passed QC. And over 1,900, so just shy of 2,000 samples have been submitted to GISAID, GenBank, and then I'm also submitting that information for each of the samples to Biosample as well in NCBI. I'm currently reviewing over 490 samples that are either in review or pending submission. And Florida has been doing really well. I have another image on the next slide that, actually, let me get into these numbers first before I say that. So the total number of GISAID samples as of November 5th is over 173,000. These are samples that are submitted from people all around the world. For the samples from the USA are over 37,000. And so Florida BPHL has actually submitted or contributed about just over 5% of the US samples, which is a lot, and about 1.1% of the global samples. So this is just to give you an idea of the number of samples that we've submitted, kind of broken up into region around Florida, just to give you an idea of, we're trying to get as representative samples to get a representative sampling from around the state. We do have a little bit more heavily focused on the Northeast region of Florida, just because, just given where Jacksonville is, and we get most of our samples, or a lot of our samples, from there. But this is just kind of broken up into the different regions. So as I was alluding to earlier, looking at the number of samples in GISAID, Florida BPHL is doing really well. We're one of the leaders in the country for submitting sequences to GISAID. We're looking at the other state public health labs that are doing sequencing. There's a few others that are doing it besides just on this list, but these are just kind of the top. I can't remember if I put like 10 or 8 on here. Florida, we rank as number 3. So we're doing really, really well in contributing a lot to the sequences that are out there. So now I'm going to get into, now that we have those genomes and that we submitted them, what are we doing with them? What is the data telling us, and what can we tell from that data? So most people are using this program called NextStrain. It's an interactive data visualization. It's like a dashboard. I'll go into it here in a second and give you a live demo. It was developed, an open source tool developed by a group out of the Fred Hutchinson Cancer Center in Seattle and University of Washington. And it's a tool that allows you to be able to build phylogenetic trees with your sequences. You can also pull in other global sequences from GISAID so that you can get contextual samples to see how your samples fit into the strains circulating around the world. And looking at the genetic diversity that is there. And so here's the link. Let me show you a screenshot first, and then I'm gonna exit out of the slides real quick, give you a little bit of a live demo, and then go back to the slides. So as I mentioned, it's a dashboard program. It's pulling in that public data from GISAID. That's why everyone's using GISAID. And the whole goal, I guess, of GISAID, and as well as the NextStrain tools, is to be able to use this to be able to have a near real-time actionable public health data, so that we're not looking at data that's 6 months old or a year old. It's something that we can look at in as near real time as possible, so that people could potentially make decisions off of this data. And so that's the whole goal of this. And it's also a fun tool that is very interactive. So let me exit out of here really quick and pull up the link. So it's nextstrain.org is their main site. If you're interested, I encourage you to go to their website. There's a lot of different things that you can play around with. I'm not going to go into detail into this, but just mention if you want to learn more about how their tool works and being able to use it for yourself potentially. You can go under the help and then docs. They have a number of different tutorials and documentation for how their tool works. And I'll just say they developed this years ago. So it's not just used for SARS CoV-2. That's just the focus of it right now. They use it for a number of different viral species, and then also for TB as well. And they have those, if you scroll down, they have all these other different pathogens that they have these different models for, but we'll just stay on SARS CoV-2 for now. So they have a bunch of different resources. If you go here to the resources, but I'm going to go to show you the main tool. If you click on latest global analysis, you'll see that it's loading. So, oh, my screen's smaller. So if you have a bigger screen, it'll actually expand. There's a map section and it'll go over to the right, but this will work for now. So there's 3 main panels or parts of this tool, I guess, that you can focus on. So on the left side, you have all the different options that you can click on, and I'll go over those in a second. So the first main thing is that you have your phylogenetic tree. So it's taking in those samples. This is a sub-sampled set of all the global samples, and basically giving you a picture of what SARS-CoV-2 looks like around the world right now. As you can see at the top here, it says it's showing just over 3,300 genomes. If you remember, there's over 100,000 genomes. I think there's now, as of today, like over 190,000 genomes. It'd be hard to visualize that many genomes on a tree in this tool. So they subsample it down to still give you an accurate picture of the overall diversity of the different strains that are circulating, but just with 3,000 genomes as opposed to over 100,000. So they have it color coded here based on the different regions. So it's really based off of continent, but there's a number of different features that you can color this by. They have clade, sampling date, genotype. There's different groups that have defined different clades or lineages that you can color them by location, country. So right now it's just colored by the different regions, and they have them colored by the different clades. So let me color it by those and I can show you. So there's 5 main clades. These are defined by the next strain group. There are other groups that GISAID has defined their own clades. There's another group they've developed a tool called Pangolin, and they are defining theirs as lineages. So there's a few different groups that are defining them in different ways, but I think next strain is kind of, I think, going to be went out maybe and be the standard, and that's what mainly we're using. But there's 5 main clades, and they have them labeled on here. And as you can see, they're color coded. The 19A, 19B originated in Asia, and then the 20 clades came out of Europe. And as you can see, as I'm hovering over, this is one of the features of the next strain tool, is that the entire thing is interactive. So as you hover over a sample, you can get the different information. So this sample here is from Australia. You can get the collection date, the authors, this is who submitted the sequence. They also give you the different clades. So this is the GISAID clade. This is the next strain clade. They have different definitions. The country, country of exposure, admin division. So in the US, that would be a state. In Canada, that would be a province. So it's based on what the divisions are called in different countries. Sometimes it's just the country name. The host, so they actually have some sequences in here. If you remember, like the news story of the tiger that got infected at the Bronx Zoo, they have that sequence in here. So it's not just, it's mainly human sequences in here, but there are some from other hosts. And then it gives you a little bit more information about the originating lab, the submitting lab, etc. So that's just a way to get more information about a particular sample. So you can also, the entire tree is interactive. So if you wanted to, as you can tell, there's just a lot of samples on here and it's kind of hard to make out different information. If you wanted to zoom in, let's say into this 20B clade, you can click on that and it will zoom in. And so as you're interested in more and more samples, if there were particular outbreak samples you're interested, you can keep zooming in there until you get to those different samples. And then there is a feature, where the zoom thing is, there we go, where you can hit reset layout and it goes back to that. You can also look at the tree. It gives you different options over here. So you can change it to a circular layout. It was rectangular. You can look at an unrooted, it's an unrooted tree, but you can look at this unrooted way, or you can look at it as a clock view here. Let me go back to the rectangular. So as you notice, this is a time tree. So the branch lengths are actually based on time, not genetic distance in this particular case. But if you wanted to look at the divergence, you can switch over and you can look at based on the number of mutations, and that changes the branch lengths of the tree. So you can see that. You can also use this slide bar here, and you can highlight, like if you wanted to look at samples from just a particular date, you can do that. Let me scroll down and show you the other panels really quick, and then I'll go back to the slides. So they also provide this map, so it maps the different samples geographically. So you can look at the different frequencies and distributions in this particular case of the clades, and it's based on what it's colored by. So if we were to color this by, let's say, country, it would color it by the country. It also has this animation. So it does it from the very beginning, from the first sample until now. Oh, I still have the, as you can see, it's going along the time ranges. If you have, let me reset it. Go to region. Yeah. You can change the speed of the animation, but essentially you can see the different samples and the different frequencies of samples pop up over time as the pandemic has gone on. So there's a number of different features here. I won't really go into all of them, but just different ways of being able to visualize this. Let me scroll down to these panels down here. So they also have this panel down here where you can look at the diversity. So you have here, this is the annotated genome. So it's a 30 KB genome. And these are the different genes or in open reading frames. So you have open reading frame 1A, B, this is S, that's the spike protein, and you have all of them. And you can zoom in so you can see more there. And then if you scroll up, so each one of these peaks, this is looking at the overall diversity, so where you're seeing mutations. So if you see different areas that have higher peaks, those are higher frequency mutation spots. You can look at it as entropy, or you can look at it as individual events, so the number of times that it has mutated. You can look at it for the amino acids, or you can change it to the nucleotide. So there's a number of different things. You can also, this is a cool feature. So let's say I'm interested in this particular mutation. I can click on that. And then now it's color coded it based on the genotype. So you can see the frequency of the genotypes. This is a great way to see, hey, is this genotype more prevalent in certain areas than others? That's a great way to be able to use this tool. And then it'll color them on the tree. So it's just a really, really nifty thing. And then it also changes the frequencies here. So since right now we're looking at the genotype, it's now changed this frequency table too. And then down here, the last thing I'll show you before I go back to the slides is you can filter by what you show in the tree. You can filter by these different categories here. So you can filter based on submission date, clade, region. I'll go down here. Let's see, admin division. Let me see if I can find Florida. Here we go. So there's 38 sequences from Florida that made it into the subsampled tree. And so if we go back up, it now highlights our Florida sequences. So you can see that all of these samples here were ones, looks like many of them were ones we submitted, as well as a few others. And then you can also submit by location and by authors. So it's a really great tool, all open source. And they've also provided a great way for you to be able to make this locally too, to make it like your own visualization. So let me go back to the slides and I'll show you how we are using it at BPHL to look at our data, since that's just the public global data. Let me go back into, okay, go to the next slide. So I just showed you kind of a brief introduction on how to explore the site. If you're interested, you can go back and play with it on your own. There's also this link here that you can go to that provides a tutorial, kind of like a blog post. It uses example data from Zika and showing you the different features of the site. So if you forget or anything, you can go to that site. So as I showed you, one way where we could use that build is looking if we're interested in the Florida sequences. For example, I filtered by Florida and only 38 samples popped up. When I made this slide, there was only like 10 samples that popped up. And obviously that's not as informative if we're interested in Florida and we want to see what does the epidemic look like going on in Florida. So how would we make a build that is focused on our Florida sequences so that we get all 1,900 or most of them into our builds? So we've made a couple different builds using the public data, just pulling from GISAID, and we can make subsample builds. So that one was a subsample global build. We can make subsampled Florida focused builds. So what it does is that it subsamples sequences from around the world, but with a focus on Florida. So we will include the vast majority of the Florida sequences into the phylogenetic tree, but then it pulls in other sequences subsampled from the world so that we can have those contextual samples. So we can see different introductions from different countries or different regions. And it subsamples those by genetic proximity to the Florida samples. So it's making sure to include the ones that are most relevant to Florida, as opposed to sequences that are not very related to us, that wouldn't be as helpful. So this allows us to get a state level look, but not in an isolated build. If we only did one with just the Florida sequences, which we could do, then we're not going to be able to infer where different introductions from outside the state came from. And so this provides a great way of being able to see that data. So we do have two different builds that are up on the internet that anyone can look at. Their NextStrain provides what are called community pages. So different groups can, we maintain them ourselves through BPHL. And so we have one that is subsampled and focused on the Southeast region. And the link is here. And so this is focused on, instead of just Florida, it's focused on the 7 states and territory in our region. And then pulling in other contextual samples. And then we also have a Florida build that mainly focuses on the Florida sequences and then pulling in those other sequences. So this is just a screenshot of our Florida focus build. And as you can see over here on the map, you can see how big the samples are. And that's not because we have more cases here in Florida than all the other states. It's because, although we do have a lot, but it's because that the tree includes more samples from Florida. But it's the same interactive tool that I just showed you, just with those emphasis on the Florida samples. So we can also make these builds for looking at data that we don't release to the public. So if we want to include additional metadata, such as county level data or city or zip code or any other thing, then obviously we're not going to release that publicly. Then we can actually use this tool using a different site to create private builds. And this has allowed us to share this tool with our epidemiologists so that they could use this to potentially investigate different outbreaks. They can add on their own metadata themselves if they're looking at a particular facility or a school or whatnot. It allows them to be able to interact with the data and look at it in a protected environment as opposed to it being on the internet. So the NextStrain group actually developed this one site here called Auspice US. And let me show you a screenshot of that. So basically, it's a website here, but you can drag and drop the final file from our pipeline, from the NextStrain pipeline, is a JSON file. And you just drag and drop it onto this website. And then it will load the build. So it will look just like the other builds, but it stays contained within the browser and the data does not go anywhere. And so this allows you to be able to interact with your data and allows the epidemiologists to interact with the data in a protected environment where it's not being shared with anyone else. And one of the other great features is that you can actually, when you have that open and on the tree, you can actually drag and drop a .csv file that has different metadata fields, and it's sky's the limit. You can add as much as you want, but you can add additional information such as county or city or zip code, as I mentioned, age or gender, or if you have disease status, any kind of additional metadata field you want, you just drag and drop it onto the tree and it will automatically incorporate the additional data into the tree that's already built. And that way, on the drop-down menu, you can then color code based on, okay, I want to look at these age ranges, or I want to look at this gender, or I want to look at individuals with this particular disease status. And then it will then highlight those samples in the tree for you and on the map. So it's a great way to be able to add in additional metadata that, for example, the epidemiologists have that we don't have, and to be able to use that tool. So this is just a screenshot of using one of the local builds. And when we incorporate county level information, now you can see that we now have these little pie charts here showing the frequencies or distributions of the different clades per county. So it gives us a little bit different information than the other builds. We can also make separate builds. For example, I made this one. This is just the example data for a particular county, if we wanted to focus in on that, because some of the other trees that have thousands of sequences on them, sometimes it's kind of hard to make out different clusters of samples that you're interested in, that you want to zoom in on. And so making separate builds or separate trees for just specific counties is kind of helpful to see. I thought this one was interesting because if you go back, something to note. So of the 5 major clades that exist, you can see in this that Florida has all 5. And we have early on, if you see, so our first sequences came from the very beginning of March. And as you can see that all 5 clades were introduced into Florida within those first week or two, and then continued to spread. And so that's what it looks like kind of at a state level. But then it was interesting just if you look at particular counties, in this case, we don't see, so we have the Wuhan over here, which is just the reference. But it's mainly dominated by just 2 of these clades. So the 20C clade and the 20A clade, these came out of Europe, versus down here you have this one sample that's the 19B clade that came out of Asia. But it's interesting, but there's different things that you can infer from this. So you can see that with the 20C clade, you had a number of different samples from, you can see that had earlier collection dates here. And you kind of had continued spread of these different clades or subclades within the 20C clade. But then here with the 20A clade, you have this one group down here of 2 samples, and then it kind of stopped. We don't have any additional samples that continued on from that particular clade or subclade. But then you have this other one with a lot more samples included that we see from a later date. So one could also infer that maybe we're missing samples that were collected, but we just haven't sequenced yet. So there's a number of different things you can try to infer, but I will get into inference and caution with inferences just at the end of the presentation. Actually, here we are. So that's one thing that I did want to note before I wrap up is interpreting these trees and interpreting this data. So as Matthew mentioned at the very beginning, once you have our samples that are collected in Florida, first of all, BPHL does not receive all of our SARS-CoV-2 samples in Florida. Many of them go to the private labs, hospital labs. And so we only get a subset. And then from there, from the ones that test positive, we're not sequencing all of our positives. We're only sequencing a subset. And then from there, we are then only submitting and analyzing the ones that then pass sequencing QC. So if you kind of see where I'm going with this, is that we don't have a fully sampled tree. No one will ever get a fully sampled tree. We never will, because we can't sample everything, and we're not able to get a sequence from every person. And so because of that, we have to be cautious with our interpretation. So I really like this graphic. This is from I did not develop this graphic. This is from the Next Gen group. But I like they provided this link here about how to read these trees and how to interpret them. And I thought this was a great example. So over here on the left, if you have a fully sampled tree, so you had let's say you had a sequence from every single person that was positive. You could make this interpretation that looking from the orange sequences to the blue, that you could infer that there were 3 separate introductions. But then let's say you were to remove just one sample, one of the orange samples, it then changes the interpretation of the tree where it looks like there's just one introduction. So very different. And then so on and so forth, as you remove different samples, you're going to get much different interpretations. So nothing is definitive. You must, when you're looking at this data, you must look at it with your epidemiological information you have as well. And always have an asterisk or disclaimer with your interpretations, just because we don't have all the sequences. So if it looks like something, if it looks like a clade maybe wasn't spreading anymore, maybe you would interpret a mitigation worked, or, maybe those people self-isolated or quarantined, but maybe not. Maybe it did spread, we just don't have those samples. So you do have to have caution with interpretation. So that is it for my part of the talk. I did want to mention one last thing, that BPHL does have a full-time position open for our sequencing team. So if you're interested or know someone who's interested in doing this type of work, you can contact Dr. Jason Blanton. He is my supervisor. He's the sequencing coordinator at this email address here. And if you could pass along that information for anyone who is interested. And then we also just wanted to acknowledge the other individuals at BPHL and our team. Our supervisor, Dr. Jason Blanton, Dr. Taj Azarian, he is our other bioinformatician. He's a part-time bioinformatician. He's an assistant professor at the University of Central Florida. And then our other sequencing techs, Omer and Jesse, different individuals that are doing SARS-CoV-2 testing in all the different sections here at BPHL, and our other members of the molecular group at BPHL. And with that, any questions? And I will see if I can stop sharing and give control back to Matthew.